we go with our second nervous system video. We're going to go through how does a nerve actually send an impulse? What is that message? The message is both electrical and chemical, and it's sent by nerve cells. So we call it an electrochemical message. It involves an electrical part of the message. The electrical part is actually just a change in charge. Polarity is the difference in charge. So I want you to think back, when you learned chemistry, you learned about a polar bond. So the reason that it was polar is that there was more of a pull from one side to another, so there was a difference in charge. One was slightly more positive, one was slightly more negative. Our neurons have a resting membrane potential that is negative. That means there's a difference of charge from the inside of the neuron to the outside of the neuron. It involves a chemical part of the message as well. So remember how there was a space at the end of the neuron? That space, the synapse, a charge can't jump a gap. So chemicals get released into that gap, the synapse, and then they get picked up by the next neuron's dendrites. All right, this whole nerve impulse is going to be called an action potential. All right, and so in order to understand a nerve impulse, we have to talk about what is a neuron like at rest. It has a higher concentration of sodium outside the neuron, and it has a higher concentration of K+ inside the neuron, and it has an overall negative resting membrane potential. So the resting membrane potential is often around -70. There's very negative DNA inside of a cell that helps add to the charge difference, but it also is really due to the fact that there's more positive sodium ions outside. All right, polarity is maintained. So in order to keep that difference in charge, the sodium-potassium pump constantly keeps sodium out, keeps potassium in the neuron, and maintains that resting membrane potential. It could be anywhere from 0 to -100, but it's usually around this -70 range. All right, next. Here's a diagram. I wanted to just show you so that you get the idea of what we're talking about here. This right here would be the axon. So this is the long part of my neuron. And here is the pump at work. If you notice these purple dots, that's the sodium. So there is a higher concentration of sodium outside the neuron. So this is outside of the neuron, here's inside the neuron. And over here we see more yellow. The yellow is the potassium. So there is more K+ inside of the cell. This is a channel protein, and it's gated, which means that normally it is closed and it's not allowing the movement of sodium or potassium in or out. That gate can be caused to be opened up by a specific neurotransmitter, and it changes the permeability of the axon and causes gates to open. Could also cause gates to close. All right, steps of an action potential. This is where we're going to really focus in on this video. How does it get from a stimulus to actually causing a change? So, first step, a receptor receives a ligand, and it causes an ion channel to open. So we've already learned about receptors and how they have that very specific shape. So if this is my receptor, and here is the dendrite, and here's the receptor on the dendrite, this would be cell body. This could be a neurotransmitter in the synapse, and it has just the right shape, and it receives it. When a receptor binds to a ligand, it might cause gates to open. There's two things that could happen. You could open potassium gates, or you could open sodium gates. If it is an EPSP, that stands for excitatory, post-synaptic potential. Post-synaptic means after the synapse. So here's the synapse, here's the next neuron that's picking it up. So after the synapse, is it exciting it or inhibiting it? To change the polarity, it's got to let sodium in. So if it opens sodium gates, it's excitatory. Enough ion-gated channels open that depolarize. So we're going to change the charge. Instead of being negative, we're going to bring it up to 0, or even maybe make it positive. And it meets the minimum value. We call that minimum value the threshold. So you have to open enough sodium-gated channels that it depolarizes the membrane and causes an impulse to send. So it is allowing Na plus to enter if it is an excitatory postsynaptic potential. Now there's another one, of course. I, think E for excited, I for inhibit. The inhibitory postsynaptic potential, enough ion-gated channels open that further polarize the membrane. That means it makes it even more of a pull, even more of a difference in charge. So let's say it goes to like -80. How would that happen? If more K plus go in. So if we have more sodium going in, oh, I'm sorry, I don't mean in. If we have more sodium going out. Oh. All right, if the potassium is going out, then it's losing some of the positive that were inside of it. So if the potassium is going out, but the sodium is not going in, it becomes even more negative on the inside. So maybe it could go from -70 to -80. That reduces the chances of the threshold being met and an impulse being sent. So this one opened the Na plus, opened channels, causing sodium to enter. This one opened the K plus channels, causing K plus to exit, go out of the neuron. All right, if the threshold is met, so once it meets that minimum value, all the sodium-gated channels open along the entire axon. So if I make a little picture here, here's the dendrites, here's the axon, here's the synapse. If enough neurotransmitters cause enough of these gates to open and enough Na plus to go in to meet the threshold, then all the sodium-gated channels will open. We call that a wave of depolarization. Depolarized because it started out at like -70 and maybe it's gone all the way to 0. So opened up all of those, letting the sodium in, changing it. Instead of being polarized, it's now depolarized. Next step, in response to the sodium gates opening, now the potassium gates start to open. So I'll make that in green. So this sodium started coming in, causes these potassium to start moving out. Well, if you've got plus coming in and then plus going out, you're going to go back to polarized. So this is called repolarization. You're going to get that charge back to -70. Now what happens is when the potassium gates open, the sodium gates close. Sodium gates are a quick shutting door, which means they close their gated channels quite quickly and stop letting sodium they stop letting sodium into the neuron. The potassium gates close really slow, so potassium keeps leaking out of the neuron, out of the neuron, out of the neuron. So instead of going to -70, the normal repolarization, it might actually hyperpolarize and go to even lower than expected because it actually let too much potassium out. I think that's my E right here. If the potassium channels close too slowly, the membrane hyperpolarizes, goes even more polarized than resting potential. So maybe it went to -80 or -90 instead of just at -70. All right, at the end of the axon terminal has calcium-gated channels. They open up, calcium moves in. Calcium has a really specific job. Calcium binds to the vesicles. So a vesicle, you remember in any cell, a vesicle is like a holding chamber. So inside, here's my axon, and here was the calcium coming in. The calcium binds to these vesicles and causes them to fuse and let out their contents with the end of the terminal. So if they release their contents, the contents are neurotransmitters. And neurotransmitters are our chemical part of our impulse. They are what is going to allow the message to go through the space and be picked up by the next neuron's dendrites. All right, recovery period. This means we're not capable of sending another message until we get everything back to normal. The ions are on the wrong side right now. Sodium went in, potassium went out. We got to pump them back to their respective sides. So the sodium-potassium pump puts them back to resting potential. No new impulses, no action potentials can be sent during this time. And continuation of impulses. In order to continue from one to another neuron, we had to release those neurotransmitters into the synapse, and then those need to be picked up by the next neuron's dendrites. All right, so I want to draw a couple diagrams, make sure that you're understanding what's going on. So here is my first neuron. Here is my second neuron. We will say this is an interneuron and this is a sensory neuron. So the receptors on here, to start this going, they pick up a message. So this is a neurotransmitter, and it changes the permeability of this axon. And what it does is it opens up Na plus gated channels, and sodium starts moving in. So we started out at -70. If sodium starts coming in, let's say we get to -50. If we're letting more positives in, this shouldn't be so negative. So it changes. If -50 is our threshold, all of these channels open, letting in more and more and more sodium. So Na plus enters, and that's my wave of depolarization. So it is depolarized right here. In response to the sodium gates opening, the potassium gates open. So I'll make them here. They would be on both sides, and remember, it's not even really a side, it's an entire tube. So around this whole thing, these are going to open as well, and those are potassium gates. K plus is going to go out. As K plus goes out, now I'm starting to lose this 0 and maybe go back towards -70. That's called repolarization. When you get to the end, there's no more sodium and potassium, so these close. Instead, there's calcium. So my Ca++ moves in, binds to these vesicles. The vesicles let out their contents. This space is my synapse. I can't have a change in charge across a gap, but I can have a chemical across a gap. That chemical will be picked up by the next neuron's dendrites. That will cause, again, another wave of depolarization. So open up the sodium gates, let the sodium happen, and let the depolarization happen, and now I'm going to have that same thing go through here. If it goes from a sensory neuron to an interneuron to a motor neuron to a muscle, it's going to cause a response. It's going to cause a change. So these are the steps that are occurring. The last thing I want to show you is how you make a diagram of it. An action potential is actually a graph of the change in charge. So over here, this is millivolts, and I start off at rest. So my resting membrane potential is right here. All of this would be membrane potential. So that means, what's the difference in charge from the inside to the outside? My resting, let's put a value on this of -70. So what keeps this right here? My Na plus K plus pump is working to keep the sodium ions out and the potassium ions in. And as soon as a neurotransmitter is picked up, if it is an excitatory postsynaptic potential, it goes up like this. So Na plus gates open, and Na plus is coming in. This is depolarization. If you meet a certain minimum amount, this is called my threshold. If you meet that amount, they all start opening. Wave of depolarization. There it is. Open, 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 open. You might get all the way to, this could be 0. The membrane potential has completely depolarized. In response to sodium moving in, potassium starts moving out. So potassium gated channels open and K plus goes out. This is not depolarization. This is repolarization. Repolarize means let's get back to a difference in charge. So, keeps going, keeps going, keeps going. The sodium channels have closed. The potassium is closing, but it often goes lower. You'll notice my negative 70 is right here. This could go to negative 80. We call this hyperpolarization. So it actually let too many potassium out. So now there's even more of a polarized membrane. It now needs to go through this. This last part right here is called the refractory, or sometimes you'll see it called the recovery phase. This is where Na plus K plus pump works to get everything back to as it was at the start. And until it is back at this point where it's negative 70 and the sodium is back out and the potassium is back in, it cannot send another impulse.